Okay, so all the molecules that are involved in the construction and the making of the cell are collectively known as, or collection, the whole category of it as will be known as the cellular pool over here. Basically similar to, you may have heard something called as gene pool, containing all the genes and everything of an organism. Okay, so like all the various genes, same way over here, all the various molecules which are present within it will be given the term as cellular pool. Now, what is the basic compounds that we have studied, especially you can say the dietary compounds that we have studied is involved in the making of it. Compounds like carbohydrate, proteins, then water, mineral. So basically whatever we are consuming is helping in the construction, in the maintenance, in the development of the cell itself. So the nutrients that we take in is basically the ones which are involved in the cellular pool in the making of the cell. All right, they are your biomolecules. Basically the thing is, they're also called as organic in a certain way, organic compounds, because they are made of just carbon, hydrogen, oxygen. So we have hmm, these groups. Then we got your all these groups together, all these organisms, all these molecules what are involved in a process that is a metabolism. So all the activities which are occurring within the cell, all the processes that are occurring collectively, we give it a term as a metabolism, which can be grouped in two parts. One is your anabolism and one is the catabolism. All right, anabolism process, generally we describe it as the process where something is being constructed, something is being made, where synthesis is occurring, manufacturing is occurring, all right, production, something to make it. Other one is catabolism, where whatever stored part is there is broken down or hydrolyzed or is broken down to its simpler form. So anabolism, catabolism in a way are opposite of each other. Anabolism is like making it, catabolism is like breaking it. So there is a constant making breaking going on in the process, and all these things collectively can be termed as metabolism. So this is just a basic in a way we are going towards a basic introduction about the cell, the constructions involved, or the molecule groups involved in the making of the cell, and how these molecules are utilized by the cell for the process called as metabolism. All right.
So we had to write it.
You okay? You wanted to write it? You can go ahead, write it. Let me know when it's done.
Done, sir.
Thank you. Okay. So about this, what is there? So now the thing is to know what it is, what all molecules are involved in the making of the cell, right? So we have to figure out, like, how do we figure it out? So we need to break open a cell and check out. Okay, just like for example, if a watch or any toy is given or something, well, you used to open it up and check it out what it is made of and try to deassemble, then reassemble like that. So we try to analyze the molecules present in it. All right. So we need to isolate it, break it apart, basically in a way like you get a computer or a laptop or something and you disassemble it and try to see what all parts are involved in the construction of that. Or another example can be your Lego pieces, all right? Something that is made up of Lego. So we see that which all types of bricks are involved in that. Will I share the slides? Yeah, you guys didn't receive the slides yet or what?
No, sir, we didn't receive.
Okay. All right. We'll check it out with Rahul sir. Okay. So now all the carbon compounds that are there are given the term bio. Well, carbon compounds, their organic content that is there generally are found within the organisms, even to a certain aspect. We can say the fossil fuel that is we have got that also comes directly indirectly from the living organism itself. So yeah. Okay. All right. Kushi also said no. So we will see. I will ask Rahul sir to share it up. Okay. So carbon compounds that are there obtained from the living organisms are given the term biomolecules. As I explained earlier, the term bio added means life, molecules coming from life, molecules coming from living organisms. Yeah, that is what it is, the simple terms. All right. The living now the thing is that within us there are the organisms that are there anyway. They are constructed of biomolecules, which can be grouped based on whether they are made of carbon, hydrogen, or not. If they are made of carbon, hydrogen, we give the term as organic. If they are made of other elements involved in it, or carbon, hydrogen is not present, we give the term as inorganic. So, anyway, majority is organic and certain aspects are inorganic constituents. So, how do we separate it or what are we doing in it? We are mixing the living tissue and grinding it with trichloroacetic acid. All right, paste it up nicely, grind it, and then what is there? Once we make that paste, then we strain it. And once straining it, we get two different parts. One is called as a filtrate, which is collected within the filter. Other one is the no, sorry, filtrate, which flows down. Another one is a retentate, which is retained within the cloth. That will be a retentate, and the other one is filtrate, which is the liquid collected in the other container. All right, and depending on that, we can say the acid soluble pool, because it got dissolved or mixed with the acid, so that's the reason it passed through it, forming a liquid. An acid insoluble fraction, which is obtained, which is left over within the cotton cloth or the strainer that is there, is your retentate. All right, from start where? Okay, Niranjan, can you explain from start?
So analysis of chemical composition. I was disconnected from there.
Not, basically, we are just trying to obtain or figure out what all what do you say, the chemicals are involved in that composition. So we are just mixing it with trichloroacetic acid. Okay. So in that we are mixing it, and once dissolved it, once like grind it and mix it, we are getting two parts. One is a filtrate and retentate, and the filtrate part we call it as acid soluble, which got dissolved in it. And the other one is all right. So we just go ahead, wait, uh, and then we group them into it. Then again, coming to identify. Okay, what we are doing basically is then once we obtain it, then we weight the amount of living tissue, followed by we after drying it we check the weight. So what we get to idea is the biomass of that, or what all mass is present in the weight. And anywho, if the sample gets burned due to the effect of the acid, then all your carbon compound oxidized pasta are removed. So any the tissue is gone due to the action of the acid. So tissue part or your carbon part is there. So whatever leftover part will be there is an inorganic content is left out. So we can then analyze it and figure out what all elemental inorganic compounds are present in it. Okay, the acid soluble part. So what we get from this is that while analyzing in this for using it, we get to see what are the acid soluble compound components are there. So what are inorganic compounds are present by analysis in it, and what all non organic compounds are found within it. All right. Now, based on their weight or based on your other processes, we can figure out or find out what all compounds are used or involved in the construction of the cellular part. Okay. Now, if you need, you can just make a note of it.
Sir, can you please show the previous one?
The previous slide, like this one.
Yes, sir, done.
Okay, I just saw the chat. And why why this acid in a per se? In a way, the interactivity of the proteins, one is in a small quantity, it can be utilized to break down, or you can say unfold the proteins that are there, purify the proteins or purify the molecules or separate them with the interaction of the ions, or you can say the polarity of this acid compared to other acidic compounds. Devangi ma'am, what do you want me to explain?
Sir, this one slide.
Which part?
This slide.
This one.
Next slide.
Okay, next slide that is there. What I'm saying is once we get it, then we check out the weight, and dry weight gives a wide drying, that means the removing the water content, we get the dry weight. So we can get what are the molecules are present or the exact composition or the compound weight that is there, the molecular weight that is there, we can figure it out. And then if the sample is burned, in the sense when you are burning it, what is left in that? By burning the sample, burning the living tissue, whatever is left out, the ash, we can perform an analysis and figure out the inorganic content in it. Because if when we are burning, the carbon compounds burn and become soot, or you can say the carbon dioxide is then gone. So whatever is left behind is your inorganic content. So we can analyze and figure out the inorganic content with what is found in them. So using these processes, using these steps, what is the aim over here? We can figure out what the, or we can get an idea of what the cell is made of. किससे बना हुआ है? What all content are involved in that? Which molecules are involved in that? So using this information, what is there? Like, okay, this is there, these pieces are there, these parts are there. So basically, it's like if you talk about a computer or something, you take your CPU unit, your motherboard is there, RAM is there, hard disk is there, chipset is there, all these things. So once you know all these, so that way we can manipulate it certain way. If we figure out what is there, if we manipulate that or exploit it to our advantage, can we construct or deconstruct a particular cell, or can we modify it to our own needs? One of the application just saying so. If we know what it is made of, if we know what it is made of using this processes, then we design something of our own. Okay, far-fetched theory, though we haven't reached there yet. Getting the idea, Devangi ma'am? So coming to these, one of the groups that are there is your amino acids. And Khushi ma'am, your query is fine, or okay, yeah. So amino acids, one of the most important nutrient, one of the most important basic unit, you can say, as these are involved in the making of proteins. May have come across earlier, all right? They're involved, they are they are called so because they have an amino group where NH2 is present and an acidic group present both, and the carbon which contains or has these groups attached to it is given the name as alpha amino acids. Okay, or the alpha carbon. So amino acids, they are the basic units. They are like those ABCDs involved in making the whole words or sentences for involved in making the protein. So if we have these, we can design a protein. Okay, if we have these, later studies we can show that if we can manipulate this, we can construct a protein. That means we can construct an enzyme of our own type, construct a drug of our own type, which I think we are pretty much trying to figure out, even like from medicinal point of view. Okay. Now basically, see, you studied alkanes and all, right? So in this, what is this trying to let me? So what is it? There is a C. So I will say this is your methane, correct? Now to this, there is one. You got your amino acid. Now you keep changing this and it keeps changing the properties of the compound. And as and when the amino acid changes and you get a variety of that. So you get your satisfied structure. That's the reason it's saying the substituted methane in the sense. Generally, methane is what I think this is what methane is. Yeah. And you substitute that. Hydrogen, carboxyl group. I think this is carboxyl and this is amino. And this R is a functional group, the variable part where you can either replace it with an H or some other functional group, maybe hydroxyl, maybe phenol, maybe benzene ring. You guys have studied about organic compounds, right? Or you are doing it in chemistry, I suppose. So, yeah, getting the idea about like how amino acid is constructed and what it is. Yeah.
Sir, can you explain the second point again?
Well, this one, what I showed you, were you able to see it? What I showed? What I'm trying to say is the amino acids are one of the functional units, or you can say the basic unit in making proteins. So amino acid, what is it there? As the name says, there is one acid group and there is one amino group. What is acid group? You have heard of acid group in the functional group. So what it is saying is now, if you take methane, what is methane? CH4, that is the molecular formula. So methane has what? Now carbon has 4 valency, which is satisfied by 4 different hydrogen. Now to this, what I do is I replace these hydrogens. I take the C again. I replace one side with COOH. I think this is how it is. And I left leave one H and another side. Okay. And this one over here is one functional group variable. That means that R stands for any other group. It can be over here, maybe attach a OH or H or CHO or add in a ring Okay, any of these can come in functional group basically. Now, since the main functional group, which is giving the name, now this is the amino part. Who asked me that doubt earlier? Yeah, this is the amino part. This is the acidic part. Since the main functional group is attached to this carbon, so then we call this as, okay. Are you guys able to see it, or do you see us some?
Yes, sir.
Okay, ठीक है, आगे बढ़े.
Sir, alpha carbon actually means when four things are attached to one carbon only, right?
Four things, or the main functional group which is participating in it, the core one. अभी अगर मैं किधर गया, तो जो अभी ऐसा है, तो ये ऐसा है, ठीक है. इधर H, इधर H, इधर H, इधर H, तो ये रिएक्टिव ग्रुप हो गया, तो मैं इसको अल्फा बोलेगा.
Okay.
वैसा, ठीक है. बेसिकली दीज आर पार्टिसिपेटिंग इन इट, तो इसलिए इसको अल्फा दिया है. Because see, if it comes in, there might be a big carbon, or you can say an organic compound attached in R. Still, I will call this as alpha. Okay. Okay. Next. Okay, I'm erasing this. So, based on the presence of different, there are 21 major types of amino acids, depending on what all alpha groups are there. You can get the list in the genetic code also in that chapter also. That is, I think, in 12th. All right. So, anyway, like the smallest one is glycine. In glycine, what we are doing is we are replacing the R with only H. So that is CH2, NH2, and COH. Or we are putting a methyl group in place of R. So we get alanine. We are putting a hydroxyl methyl. So we get serine. So as we are changing it now, replacing this, or as we are changing this R group, the whole conformation or the property of the compound is changing. Property from the perspective of charge, from the perspective of its pH, whether it will be remain basic, whether it will remain acidic. There are, okay, just don't go by that, okay, just mention acid to acid ही रहेगा. All right. Charge changes, negative positivity. On that basis, because of so much of change occurs due to the presence of these amino acids, and each amino acid's property is can say very heavily contributes to the structure of the protein, that even a slight change, by chance if an amino acid has made up of many alanine and I change it one aniline with alanine with glycine, there is a lot of structural change in the protein, and which can create give me a completely new protein structure. So that is the, you can say, the importance in a way, or how significant amino acid plays a role in the construction of the protein. Not just construction, even its stability, even its shape and structure. Because of the presence of these different amino acids, the enzymes. Have you come across enzymes before? Not much thoroughly. Okay, we will see. So enzymes that are there, they also get the specificity. All right. Now, number of amino and carboxyl group, these are acidic, basic, neutral. All right. So we get different properties, different glutamic acid, basic, lysine, valine. So now what is it showing is that if we take valine, even though it's an amino acid written there, acid, but then it shows neutrality from the pH scale. Lysine is showing more of towards the basic side, alkaline nature, and glutamic acid, well, as the name says, it's acidic. Okay, if you want to make a note, make a note. Was in the cell, as in they will be found in. They are found in the making of the protein, and a protein is found in certain aspects of the cell membrane, cell wall. Cell wall is peptidoglycan. Yeah. Well, what you can guys can do is just take a screenshot of this where it is just showing the different types of amino acids. All right, the ones that are there, your basic, uh, acidic. All right, then depending on the polarity, the charge. So, though you are not completely expected to by heart and remember all of this, but you are expected to know that these all categories are there. मुझे ऐसा एमसीक्यू नहीं आएगा कि लाइक ये बताओ ये स्ट्रक्चर है ये पोलर है क्या नॉन पोलर है. Okay, we'll proceed ahead. All right. So we got similarly the aromatic amino acids as an aromatic compound. The functional groups are aromatic in nature. So there is a tyrosine, phenylalanine. Now the ionizable nature that is there is due to the presence of the acidic and amino group. All right, the acidic group and amino group. So because of the presence of the functional group and with the amino group, acidic group, the pH on the structure variation comes in. Now these that are there makes zwitter ion which carry both positive and negative charge. So in this case, when they're interacting, this amino group and COH group provides the charge. As a result, it is called as a zwitter ion. The plus and minus, both the charges are present. And the interaction of amino acids with other amino acids results in a bonding pattern, which then helps in construction of the protein chain. Now, in this is the general, you can say the structure. Now, this can be charged in a sense. If H is 1, this would be, I think, minus. And if I add H over here, this will become plus by adding a proton. Yeah. This state that is there, wait, let me rephrase this part. Okay. This state is what is called as a Zwitter ion. So there is positive, there is negative. So the amino acids have the ability to form this. Yeah, you can say it to a certain extent, like kind of salts. Okay, so they can form this. Now, another aspect is that once they are this, they can interact with another amino acid and bind to it. Then that is a different bonding pattern. They form bonds in that between two amino acids and form chain and links. Now, coming to okay, Kushima. See, polar, non-polar, meaning that polar means there is polarity. That means some charge is formed in it. Non-polar means it is not forming any charge. So, like, how do we say? Uh, it is not ionizing in a way, right? Not letting form any ionization in it, no ions formation. So that we can say is non-polar. All right, and the one that is uh, polar means there is polarity in it, like North Pole, South Pole. Charges are developing in it. So electrically charged compound, exactly, you will not say it is showing any potential, like shooting out electricity. Electrically charged in the sense there is polarity is involved. So since polarity is there, if any charges are there, it can pass on. To a certain extent, you can also use water. If it is interacting with water and forming polar in it, that means that compound is polar. That means charges, ionization is occurring. If it is non-polar, it will not interact with water. Okay, we can use the water also as an example to figure out whether it is polar or non-polar. I hope this in a way clarifies your doubt, Kushi, what polarity means. So in a way there is polarity, like, yeah, poles, that is where charges are present. A charge in the sense positive, negative interaction is occurring. Okay, that's a Zwitter ion nature. Oh, yeah, yeah. So this is a normal amino acid. This is with Zwitter ion formation having partial charges in it. Again, last slide. This one. Now coming to another group which are called as the metabolites. Now which are having an integral role, the molecules which are having an integral role, direct involvement in the growth development of the organism or the processes that are followed within the organism, they are given the term as primary metabolites. Without them, the organism cannot go ahead. In addition to this, there are certain metabolites which are there playing an important role, all right, but they are not directly involved, so they are called as secondary metabolites. Okay, they are there for the organism, but even without it also may do ahead. And in your CBSE textbook, there is a list given. You may have to remember that because sometimes a direct question may come in the NEET and all. Like which of the following is not a secondary metabolite? Which of the following is a secondary metabolite? Okay. Now, some of the components that are there which are produced in it, you can say, for example, like certain organisms produces this antibiotics and all, which are required by it's like that is helping the producing cell to ward off competition with the food, and it becomes useful to us because we can help in the medicine part for curing disease and infection. But then it is not directly involved in the survival. If that secondary, if that antibiotic is not produced by the cell, it will still survive. It can still be it as it is, and the cell process will go on. So primary is the one which are required must. Without it, the organism cannot do. Secondary is like additional products are formed for its benefit. And what we as a humans have found is to use it to our advantages. Right? That is a secondary and primary metabolite. Okay. Yeah. Can I change it? These are just some examples which are anyway there in your textbook as well. So it's not compulsory that you need to write it, but then you need to know the names. Functional point of view questions will not come, like what is it used for or what is there. But then from the term point of view, questions can be asked like given you a list. Like, okay, carotenoids, morphine of lemongrass and in that then certain other products will be written in there, compounds written in there. So you have to say, okay, odd man out or of the list, how many are secondary metabolites, how many are not, such kind of questions. But then questions are not expected like to remember what is the use or what is the function or what they are used for. Okay. These you will find in your text TB that is there. You can open it up and check it out. So just highlight them and keep it. Next. Now, what all biomolecules we have in the cell? One of the important one that is their carbohydrates. Well, you have come across carbohydrates earlier many times, made up of the elements your carbon, hydrogen, oxygen. All right. So hydrates, that is your, well, the word means the water joined with carbon in a way, and they're also called as saccharides. Saccharides are sugar, all right, sugar molecules. That means certain compounds, certain carbohydrates are sweet to taste. So the elements that are involved, the main elements that are involved in its construction are the carbon, hydrogen, oxygen. Okay, and they are in a particular pattern, and they are there. So general formula CnH2n for simple, but then we generally don't follow this. are not exactly having this formula for everyone. Generally, we follow okay, this is the general formula generally we follow. This is as per your text. Okay. Now, organic compounds like formaldehyde, lactic acid have the empirical formula, that is these parts. The formula is there, but are not considered carbohydrates, even though they are satisfying the need of certain elemental formation, but they are not considered as carbohydrate because of the certain other chemical properties, difference in the chemical properties. Okay. Oh, yeah. Please make a note of this formula. So the carbohydrate molecules that are there, the functional groups we can see in them are either aldehyde, presence of a ketone group, or presence of hydroxyl groups in them. Okay, so now these three groups we can find them, and majority of them have definitely have the presence of hydroxyl group. Now again, the categorizing them based on whether they can be hydrolyzed further or not. Okay, if they cannot be further hydrolyzed, that is the molecule that is formed is having these groups, and once we hydrolyze it or break it down, so these groups are breaking down and some other compound is forming, then the end one that is there, the basic unit, or the one that will be satisfying the properties of carbohydrate, the smallest one, the basic one is a monosaccharide. So mono is one saccharide sugar molecule. All right. So if they are further, if they are broken down further, they will not be carbohydrates anymore. So they are depending on the presence or absence of which type of groups it's there. That is either aldehyde or ketones. They can be grouped as aldoses, ketoses. Hmm. Now, generally they have your 3 to 6 carbon atoms, like 3 carbon atom monosaccharides can be called as trioses, pentoses, 5, tetroses, hexoses, like that. Okay, which are, and some are crystalline, some soluble in water, and many of them are sweet to taste. So because of this sweet nature, we give the name as saccharides, the sugar. All right. Eighth one. This this is from a textbook. That's the reason I didn't go further in that. It's like the list is given. It's just been. You can take a screenshot of it if you want. List.
Sir, morphine is a drug, right?
Yeah, very good one. Okay. So further ahead, what is glucose is type is a monosaccharide which is made up of 6 carbons, so it's a hexose. All right. Now, glucose, as we all know, that it is one of the important molecule in our body. It is one of the substrate through which we use the process of respiration to generate energy, and it is required in certain quantity in the blood. If it is too high, very bad. If it is too low, very bad. We need an optimum quantity found in the blood to run the whole system in a proper way. Right. Now, it's because of its structure, the size, and all, it's able to pass through the cell membrane easily. It gets utilized very fast, right? and the energy is utilized as mentioned earlier. So that is the property of the glucose. Then another one, that is a galactose. Now, alpha beta that is coming in is on the basis of the position of the hydroxyl group in the cell. Okay, of the hydroxyl group in the functional carbon. Now, galactose, another type of monosaccharide, which when joined with glucose can give lactose found in milk, right? Now, though they are similar, but then we cannot like interchange them. We might make the glucose and a galactose, or galactose to glucose. Though even if galactose is there, glucose, now due to their structural part or the position of the hydroxyl group, uh, okay, so you 1, 2, 3, 4, 5, 6 carbon. So this is a structure given a general structure of glucose. Now, if the OH is present this way, we call it as alpha, and if the OH goes the other way, then we give the term as beta. So depending on the arrangement of the hydroxyl group with respect to the bonding pattern or then this ring structure of the compound, we give the term as alpha form or beta form. Okay, this is the difference comes in position of it, and this position plays means just like a small position, whether it is like in a perpendicular structure or in an opposite way. So on because of this presence, the whole you can say the property changes, the interaction with the other compound changes, their reactivity changes, whether how they are stable or not, their bonding pattern will change. I hope this gives an idea. All right, it's just a small change, but this is what the alpha beta form is given again. This is your alpha, this is your beta. Structural isomers of itself, to us, you can't call that, but then the thing is, yeah, you can say so. The molecular formula is changed, same, but it's about, I won't say exactly isomers of each other of glucose. It's just, you can say a variety of glucose found. If we say another aspect that changing, you can call the structural isomers, we can use the term for glucose and fructose, right? They are isomers of each other. Okay. Now, Niranjan. No, it is not galactica, it is galactose. Okay, what I'm saying, what is this? These two, that is galactose and glucose, they are a type of monosaccharides having similarity in the content of the carbon, hydrogen, oxygen, and having an existence of two forms. Now, When galactose plus glucose join, they give us another type of carbohydrate, that is lactose. And interchanging between the glucose galactose is not easy, and we cannot use galactose in place of glucose for the respiration process, again due to the arrangement of the functional groups, right? So here is the three structures given. The three, one is a fructose, one is glucose, one is galactose. Now we can say that they are all, as Kushi ma'am said, structural isomers. Now the formula of glucose fructose is same, but structurally you can say they are different. Whereas structural and galactose and glucose are also in a way isomers again, right? But functional group's position results in changing of the property. Ribose, no. If you count, okay, Prathamesh is the second one ribose. No, it's written the fructose. See, there are one, array, yeah, yeah, yeah. If we talk about this middle, this is the first carbon, or you can count like this: one, two, three. All are having six. Ribose is, I think, made of five carbon. Okay, monosaccharides, all of you. Now, fructose name comes from the part that it is mainly observed or found in the fruit. Fruit as like a fruit sugar. It is called as a fruit sugar because it is observed to be in the present in the fruits. All right, and it is a ketohexose having the ketone as a functional group. And in the ring formation, it is pentameric than hexameric, what we observed for glucose galactose. Now, fructose and glucose are structural isomers. They are similar and they are interchangeable. All right, they can, like glucose can be changed into fructose, fructose can be changed into glucose. And when fructose and glucose join, we get your sucrose, the sugar what we use, consume, table sugar. Disaccharides that is there is when two monosaccharides join, right? Mono is one. So when Two sugar molecules are joined clubbed together, chemically bonded with each other. That results in making disaccharides. Yeah. And they are formed by glycosidic linkage or glycosidic bond. The bonding form name between the two monosaccharides which are participating and joining to form the disaccharide is called as glycosidic bond. Disaccharides generally are sweet, crystalline, easily soluble in water. Like for example, classic example is a sucrose, right? What we use as a sugar. Then another one that is lactose, which I think we find in milk. Then another form may have heard of maltose. If you guys remember, like you consume Horlicks, Bonvita, or Maltova, that contains it, right? So sucrose that is made up of glucose plus fructose. Non-reducing sugar means that it is not getting reduced when we do a particular test. Uh, that is called as Benedict's test to identify whether the sugar is found in it or not. Or all right, now then lactose, again glucose plus galactose, so it gets oxidized, so it is reducing. Then maltose joined by two glucose together. These are the examples of disaccharides. Here are the structural representation of the disaccharide. I think I'm facing some issue. Okay. Now, what we see, this link up that is occurring, all right, this link, and this link, This link is called as a glycosidic bond. Okay, formed by condensation reaction, basically like water is released. So this link that is occurring generally is found in the first carbon and in the fourth or fifth carbon. Okay, so like that. 1,4, 1,5 linkage, 1,4 linkage. No, sorry. This is your 1,2. And this is your, so like that. 1,4, 1,5 on that basis. 1, 2, 3, 4. This is your 4. This is your 2. This is your 1. In this case, 1, 2, 3, 4. 4, 1. So the linkage that is occurring is between your 1, 2 position, 1, 4 position. Or it can also occur between 1, 6 also. Okay. So this linkage that is occurring is glycosidic bond. And if such link, another one comes in over here, then it will be this, what we call, this is what, disaccharide. 2 are there. So if another link occurs over here, then it will be 3. And another one over here, then it will be 4. So depending on how many monosaccharides are involved in the construction of the bigger carbohydrate molecule, so this will be called as trisaccharides. This can be called as tetrasaccharide, 5 penta, so on and so forth. Okay, penta, hexa, septa, octa, like that. So that is how it has been bonding, and this reaction that is occurring is a condensation reaction resulting in formation of the glycosidic bond, or also called as glycosidic linkage. Okay. Before we go into this polysaccharide, now generally from your two molecules of saccharides joining 2 to 10, that is the 2 to 10, is given the term as oligosaccharides. All right, 2 to 10. Beyond 10, that is 11, 12, 13, 14, so on and so forth, 100, 200, 300, we give the term as polysaccharide. Technically, as soon as this disaccharide starts, that's a polysaccharide because multiple sugar molecules are added. But we, for our ease of understanding, for our ease of classification and information knowledge point of view, we have made a subgroup, that is a group called oligosaccharide, where generally we consider the multiple saccharides, multiple sugar molecules, multiple carbohydrates that are joining in is from 2 to 10. So your disaccharide, trisaccharide, tetra, penta, all these will come under oligosaccharides. And beyond that, whatever is there, we will call it as polysaccharides, where many such glucose or fructose or galactose or ribose, whatever monosaccharides are there, are involved in the construction of a large carbohydrate molecule. Right? I hope you got the classification, what I was trying to explain. So coming to this polysaccharides. So poly means many, like we use the term polyethylene, polymer, or polygon. Okay, a geometric figure. So many sides, that is polygon. So poly, many sugar molecules are joined, that is a polysaccharides. So forming a large chain of molecule, either same or different. Okay, they are generally amorphous, not having particular shape. Then tasteless, will not find any taste, and generally insoluble in water, or very slightly, or it's like they will not easily dissolve unless hydrolyzed. Is my voice coming in now? Okay, yeah, so that is what the property of polysaccharides. Well, these are like the easy example is your Legos. You join the same unit one after the other and make a big piece. So you can deconstruct them, that is, you can break them into or separate them into smaller single units, that is, a monosaccharides. So if a polysaccharide is there made up of, say, glucose, galactose joined together, or glucose, fructose joined together, so you can separate them by the hydrolysis reaction and break them down into monosaccharide. Uh, if you remember your last year, that is on 10th life process chapter, digestion and nutrition, where you studied about enzymes which, like, I think salivary amylase, breaking down the starch when we take the food in the mouth. So starch is an example of polysaccharide. Due to the work of the enzyme, it is hydrolyzed to its monomeric units towards glucose. Okay, so that is what that it can be broken down into its units, what it is made of, and during using condensation reaction, they can be reconstructed. So basically, in our body, that is what happens. We take the plant stored form of carbohydrate starch, consume it, it gets broken down by the enzymatic activity into its monomeric units. Then our body reconstructs it, joins it into the animal storage form, that is glycogen. So so it's basically like reassembled, reassembled. Now, depending on the bonding, depending on the size, depending on how many branches are there or branching is there or not, the property of the polysaccharide will change. Some polysaccharides are easy digestible, some polysaccharides are not. Like starch is easy digestible. Glycogen is polysaccharide. Glycogen starch similar. Glycogen has more branches compared to starch, and starch is a polysaccharide. Like cellulose is a polysaccharide. Now, starch, what we when we consume, it's easy digestible. Like as soon as you take the rice which contains starch, it is broken down in the mouth. Starts the digestion starts in it, and the body goes ahead. But the cellulosic part that is there of the plant when we are consuming plant doesn't get digested. Well, our body is not equipped to digest it basically. Yeah. So our body uses it as fibers for bowel movement. So now cellulose again is a polysaccharide, but due to the arrangement of the monomeric units or the linear chains that are there, its properties changing. All right. That is what this means, that on the length, on the branching pattern, the folding, coiling, if at all is there, okay, or layer after layer, how they are held together. Yes, that is another reason we don't have the enzyme to digest it, to break it down. That is also a fact. Yeah, Kushi, yeah, that is also there. We used to. Now, due to the changes in our eating habits, we don't digest cellulose. So, about polysaccharides, moving ahead, starch. Let me see if I can show you its structure or not. Okay. Okay, let me just show you a zest of the disaccharide and polysaccharide, the formation. Wait. Can you guys see this? Okay, this is about disaccharides as we were speaking. All right. So in this, the bonding pattern that is there, what I was discussing. Yeah. Now, okay, this is the structure of alpha and beta glucose. So the beta and alpha nature that is there, as I was saying, is due to the position of your OH. See over here, with respect to that. Rest all remains pretty much the same. So due to this change in the position of the OH group, the whole position, everything changes. All right. And this is the condensation reaction occurring in it. So you can see over here is the fourth carbon and the first carbon between the glucose galactose, leading to formation of lactose. Same over here between the first and the fourth carbon. Oops, 1, 2, 3, 4. Same thing over here between glucose and fructose. What we have got is the first and second carbon. Though I haven't come across questions in your case where they're asking about the beta or alpha or like that, which is used. But yeah, the bonding pattern definitely gets asked. What type of bonding is there? Like which carbon positions are involved in the bonding? Okay. Now, in the case of this saccharides that are there, formation, that is the glycosidic bond as I was explaining. So there is a condensation reaction occurring between the first carbon and the fourth carbon in this case, leading to formation of glycosidic bond. So 1,4 gives the idea to us that which carbons are involved in the construction or in the which carbons are involved in or participating in forming the glycosidic bond. Now this is reversible. If we are doing condensation, it is joining, and when we hydrolyze it, react with water, we can separate them. Okay. Now by joining these together in multiple ways, what we get is polysaccharides, as I was explaining earlier. So starch that is there is one of the form of polysaccharide, which is again made up of your amylose and pectin. Okay, so going back to this, coming to the starch. Now in this case, what is there is a stored form of food. Now starch is the stored form of carbohydrate found in plants, where the amylose and amylopectin made up of alpha glucose is there involved in the making the starch. Now in this case, Yeah. Now, amylose that is there is branched. So in this, the amylose that is there is like same units are joined together in a straight chain, but then to stabilize itself, we find that they're in helical pattern. But there is no branching. That means it's a straight chain. It is unbranched. It's a straight chain of glucose, alpha glucose joined together and without any branches. But then to stabilize itself, we observe that it is helical in nature. Whereas the amylopectin that is there is a branched polymer. Okay, in it we may observe these subunits of glucose joined together, and from here another branch will take place. This branching that is there will join with 1,6, that is sixth carbon atom of this one of the main chain. Okay, so this will show you amylose will show only one 4 glycosidic bond, whereas amylopectin will show one 4 as well as 1,6. This 1,6 is the connecting link for the branch. I hope you guys are getting the point. Now, glycogen is also made up of glucose. Difference comes that it is heavily branched. So this is there is a straight chain which is coiled. This one is a straight chain with branches coming at a distance, whereas glycogen that is there is a straight chain with more branches. If there is a branch over here, then another branch will arise from there, another branch. Okay, like this. It is also involved with glucose molecules are involved in the same way for its construction. And similarly to amylopectin, branches are there. At the branching point is your 1,6 bonding of the glycosidic bonds. Okay. Now, difference is that starch is the storage form of carbohydrate in plants. Glycogen is the storage form of carbohydrate in animals. Particularly, it is constructed in liver and stored in liver and muscles. Whereas and when required, it can be broken down and consumed. All right. Uh, you are asking or telling? Okay, then you can find glycogen in microbes also. So in fungi, bacteria, yeast. Basically, it is like a reserve for energy. As and when required, it can be broken down to release glucose. Yes. So this over here is showing the straight chain with 1,4 linkage. This over here is the linkage of a branching pattern forming with 1,6, same as 1,6. Now in this diagram, this is non-reducing end because this is the one with a carbon which is participating in it. So since the carbon that is your anomeric in the sense the one bound to the oxygen is free, so we can say that this is reducing end. Here it is not. Here it is not. Same way, this side is reducing, so it will help in oxidizing or participate in your redox reaction. This will not be able to. So the saccharides, which who has this end free, are considered or will have majority of it to be able to interact and participate in the oxidizing reactions. So they will be your non-reducing sugar, which is not able to will be your non-reducing sugar. Okay. Now coming to cellulose. Um, Wait. Can you please explain 1,4 and 1,6? See in this. Now this is 1,4. Okay. Now if I draw this, so this is the first carbon is present over here, second here, third here, fourth here, fifth here, and CH2OH. This is sixth. Okay. So when this link is forming, you can see a 4 and 1. So the first and fourth carbon is participating in the glycosidic linkage. So that is called as 1,4 linkage. Okay. And since then in the 1,6, this is the sixth carbon. This is the first carbon of the other branch. So for example, say this end is involved in bonding with the sixth carbon of this one or something like that, or sixth carbon over here. So this will be a first carbon of this and sixth carbon of the one already there. The thing is that over here in this line 1414 is booked, and this chain first carbon is there to react but fourth one is not there, and this is a free carbon which is away from the chain ready to interact, so it comes and binds to it. So this link is your 1,6 linkage. Okay, that means 1,6 means first and the sixth carbon. 1,4 means the first carbon and the fourth carbon. Now coming to next, that is cellulose. Cellulose, another form of polysaccharide observed in plant cell wall construction of the cell wall of the plants. Okay, the primary cell wall, helping in protecting it, helping in giving it the rigidity, helping it the shape. Okay, it is one tough one and well not easily broken down. Even if the cell dies, the cellulose layer remains, helping in making of a tissue called a sclerenchyma. Right. Now the reason is that it is tough due to arrangement of the chains. Right. In it, what we observe is alternative bonding pattern between the units, which gives it the strength. Secondly, it is unbranched, straight, but layers, that is the linear chains, are connected to each other by hydrogen bonding. Okay. Now in this there is the units that are there because of the beta glucose that is present. What happens is the bonding pattern that is there is alternative. So as a result, what happens? They don't coil. It is not branched. There is no branch coming up from here like this. It is straight, but because of the alternative bonding, that is the pattern of the bonding or glycosidic linkage that is occurring between the two participating beta glucose, it does not coil like amylose, what we saw previously involved in making of starch. So it remains straight. Now, what happens in this straight pattern? Then the another chain parallel chain is found Okay, so in this, again, similar way, what we observe another thing about between these two chains is that there is formation of hydrogen bond or hydrogen bonds between the two layers or between the two chains. This edge bonding gives it an extra stability and added strength, helping in maintaining the rigidity and the turgidity that it shows when the cell is exposed to certain stress, plant cell. Okay, that is where cellulose also becomes quite a strong polysaccharide. Okay, break. Okay, you all want a break? All right, fine. In my watch it is 6 9, not 6. Sorry, 7:40. So I guess. Oh, you want a quiz? I'm not sure yet. Let me check. Okay, I'll give you the break, and when we return that time, we will start the quiz. Okay, it is 7:40 now, correct? So 7:55 is fine. So going ahead with the other. So there is another carbohydrate, inulin, found in the plant. All right, we generally find it in the plant. It's a type of polysaccharide generally containing made up of fructose units joined together, generally found in your plant roots, the stuff where it is used as a storage form. All right. Now, in general, about polysaccharides, as I was explaining earlier, one of the end is reducing end, where the carbon, which is connected to the oxygen that is part of, okay, in the structure, which is called as atomeric carbon, that is your reducing end. If and the other end, which is not connected to it, is called as non-reducing end. Okay, that is part of the basically in this part. We take it this way. So this is your anomeric reducing end, and this will be a non-reducing end. All right. So two ends. Then, and generally, when we draw this, we see this as the right side, and this as the left side. Okay. So that is that. As I was talking about the alternative bonding pattern due to the presence of beta glucose. So this will then like this. So one is up, one is down. So we see such patterns. Because of this arrangement, this will not happen. The helical pattern will not take place, will remain straight. So what will happen is another layer will come under this, another one lying under this, and they will have hydrogen bonding. So that will increase the strength of the cellulose. So more different types of polysaccharides are there, bound with amino group like N-acetyl, galactosamine, glucosamine, all these things. These are involved in formation of, oh, okay, which one you want? This or this? Okay, I was go with this. No, 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 it is inulin. Insulin is a protein. Inulin is another type of compound. Okay, it is not insulin. Please, 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 when you read question comes in by chance, if you are like any unfortunate event, if a question comes on this, please, okay, yeah, if any question comes, remember that this is a correct term. It is not insulin. That is different. Okay, please make a note on that. This is a carbohydrate found in plant. Insulin is found in animal, especially mammals. Okay, which is a protein. This protein. This carb. Okay, okay. Okay, this is a structure. Somebody asked about structure, I think. No, slide before the diagram. Okay, here you go. One thing is that there are other many different types of carbohydrates you will find, many different types of polysaccharides are there. Okay. Now, if the polysaccharides that are there made up of the same type of monomeric unit, same type of that is a monosaccharide is involved in joining it, then it is called as homopolysaccharide. If the monomeric units that are there are different in it, like glucose and something else is there like that, then it is heteropolysaccharide. All right. So another polysaccharide is chitin. This is again a homopolysaccharide. It is observed. It is a part of your exoskeleton arthropod. Then it is also part of the cell wall of the certain organisms like fungi. Okay. So homo, as we have discussed before, same. All right, same. If something is different, that means homo in the sense they are made up of the same monomers. That is, the basic unit is repeated. In the case of hetero, it is not. Okay, it is something else. Like for example, your hyaluronic acid or peptidoglycan. These are the polysaccharides made up of a different ones. Do you want to note it down? So this is the structure of your N-acetyl glucosamine. Joined together. So to a glucose and amino acid is acetyl group is added in N-acetyl group. So monomeric unit. And this whole bracket with N is like multiple units of that. This will help in making chitin. Chitin. Just write the spelling correctly. If you want to take a screenshot of it, just take it in. So this N that is there, that is repeated units. Okay. Now coming to the uses, what all role is there? What is the main with respect to our need, with respect to cellular need? Energy, right? Main source of energy, a primary source of energy, you can say. First thing that the cell to obtain energy for its work will go for carbohydrate if it is present, right? Now, then one of the carbohydrate, that is a glucose, will form the main substrate, the main base that is involved in the synthesis of ATP. That is ATP. I guess you all know what ATP is. So it will help in making that by participating in a process called respiration. Then lactose is present in milk, which is again acting as energy source. Okay. Though now in our body there is an enzyme. Okay, that is irrelevant. We will come to enzyme lactose. Excuse me. Good. Then in certain cases it is acting as structural component that is in the making of the cell wall. Some glucose might be involved in cell membrane formation also. Okay. Then reserve food. So main source is energy. That is the primarily it is used as an energy source. If it is in excess, it is used as stored form, starch, glycogen. Then involved in the construction of the and found as a membranal part, cell wall. Then also in this food source, in something like milk and all. And mainly the energy that is there is for the metabolism is obtained in the form of energy ATP by breaking it down. So where glucose is the main substrate. Okay. Now coming to the next one. proteins. This is another type of biomolecule. Now, the biomolecules that we are dealing now with, carbohydrate, proteins, and all, they are the macro ones. You can say macronutrient in the sense like they are needed in quite higher amount for by the organism for its functionality as a nutrient. And also they are larger in size, especially the poly ones, the polysaccharides that are there. Okay, so proteins. Anyway, what is proteins? Now, proteins are long chain of amino acids. What we saw earlier as a small molecule called amino acids. These are clubbed together, joined together, just like we saw the monosaccharides, which are joined together to make polysaccharides. Similarly, amino acids are joined together to make proteins. Before we proceed ahead into proteins, try these questions. Uh, okay, I got one answer from Niranjan. I hope this screen is visible to all. Himansh B, Devangi A, 1, 2, 3, all. Okay, any more takers? Jay C, okay, Niranjan C, Shreya C, Rashmi C, Khushi C, Dipendra C. All right, let's see if the majority wins or not. An audience poll. Ooh, yeah, it is. Good. Let's see. You can see here it is made up of straight unbranched chains which are parallel, kept parallel to each other and packed together held by hydrogen bonds. Now, ionic bonds are not a feature of carbohydrates, neither lipids, which we will see later on. One of the bonds found in proteins, which we will see afterwards. Okay, so if you want to take a screenshot of this, then I will go to the next one. So majority of you got it correct as C. So yeah, you were paying attention. Good. Now let's see about this one. Uh, saying B, D, C, D, C. We're randomly just hitting around with almost all are there. Let's see. So what I see is majority of you are going for D for Dubai. Okay. Nice. So it is D. As there are branching and it is made of alpha glucose. So the bonding is alpha. And here we go. Okay. So 1,4 glycosidic bond 1,6. That is also the alpha glucose is involved in the making of it. So hence B cannot be correct because it is only 1,4 alpha. A so definitely not because it is beta is involved. Well, C is saying 1,4 beta and 1,4,6 alpha, which is also incorrect. So ultimately left out is D. Now, which is the molecule below is B glucose. If you guys remember from the slide I had shown earlier, this type of question, I guess, rarely will come, but anyway. Let's see. Yo, B again. B for Bombay. So difference between alpha and is the arrangement of OH and H attached to the first carbon. That means it is only in this region. Rest all is same. So if this is beta, can you make a guess on of the four which one will be alpha? Absolutely right. Yeah, it would be D. So if B option is of beta, then alpha will be D option. Okay. You can take a screenshot and you can take this, the last 2 points that are there is describing what is the arrangement of the OH group with respect to the carbon attached to the oxygen. Yeah, okay. Can you make a guess? Okay, this question, they have given a molecular structure, all right? They have given a molecular structure here, and this is how the bonding pattern is. So this is a chain, the bottom one, where my pointer is hovering, and above there is another chain which is connected to it. Now they are asking From these four examples given, amylose, aminopectin, cellulose, glycogen, which of these four can have this given image pattern on the figure that is drawn for the polysaccharide? Make sense? I hope you all got the question. Okay, you are. And okay, one thing, understand this, the diagram shown there is branching. So try to remember which all polysaccharide we discussed has branching. Okay, let's see. Okay, remember amylose is not having a branch, it is forming a helix. Cellulose is not forming a branch. So 1 and 3 is out of question. So C and B and A gone. So what is left? 2 and D is left. Last question. Okay. Back. Okay, so next one that is there is your protein. In this case, what are they? They are an important group of, well, as the it says, important group of biomolecule. All right, more than 50% of the dry mass is containing protein. Now, one of the statement that is there, this one, all enzymes are protein, but all proteins are not enzyme. Well, have to remember it, etch it in your mind in some or the other way. And proteins are also, may have studied in lower class, they are the building blocks of the organism, for helping in growth, helping in development. Okay. Yeah. Here is it. Okay. Now, the proteins essential part of where are they useful? Then cell membrane, they are there because that contains your phospholipid bilayer and integral proteins in the membrane. Then hormones. Over here is you see insulin. This is a hormone made of protein. Inulin is different. It is a carbohydrate found in plant. Right now, important proteins like carrying oxygen, hemoglobin, which you have heard many times with studying blood. Then myoglobin is part of muscle. May have come across in earlier chapters of biology. Very good. Myoglobin part of muscle. These help in carrying oxygen, right? More the myoglobin a person has in the muscle, more is the red color the muscle appears. So antibodies that are there now, you may have heard of antibodies a lot with respect to this corona COVID-19 and all that comes in article. These antibodies that are there, they are also made of protein, all right, which is developed by our own immune system to fight against, to ward off against the infectious agents. All right, then your nails, hair, well, they are made of protein. Actin, myosin, again, are proteins which are involved in your contraction, the muscle movement. And so all these proteins that is there is made of the basic unit which we saw earlier, amino acids, which will come across again. All right, the same monomer unit. Now there are nearly 20 different amino acids that we are dealing with, and their combination results in formation of infinite number of proteins. All right, you mix in 5 amino acids, so 10 amino acids or something, you get your protein. You make hundreds of amino acids joined together, you get your protein. And they can be repeated units, non-repeated units, alternative, however you want to do it. Combination is nearly infinite. Now, amino acids, as described earlier, the central carbon atom following the alpha carbon atom connected to a carboxylic group and an amino group. All right. One is hydrogen, the other one, if hydrogen, it's a glycine, or there can be a functional group. So, so this, if we keep changing this R group, and we get a new amino acids. So we'll just proceed ahead. So you can see again an example given: lysine, H, alanine, methyl, leucine. I guess it is butyl group. So you get your different amino acids. Now, what is making the protein? What is the basic construction? Two amino acids combined together bind with their amino group and the other's one's carboxyl group. Okay, this bonding, this attachment results in formation of a bond called as peptide bond, peptide linkage. Again, water is released. Okay, it's like condensation reaction. So water is released, and as a result, a bond is formed. So this bond is peptide. This is the basic hold. This is the joint which many of these help in making the protein that we know. So this bond is peptide, and many such peptide bonds link many such amino acids together, forming a long chain. So many peptides are there. So this thing that is there, this unit, is what we will call as one peptide. And when such peptides join, then we will call it as polypeptide. That is what protein is. So protein basically is made up of many peptides joined together, and each peptide has two amino acids bound to each other by peptide linkage. ठीक है, आगे बढ़े. So called peptides, any number of extra could be added to the chain in any way, and they are called as polypeptides. So the protein molecule may just be have one polypeptide chain, or it may have two or more. So basically, it is a long chain of peptides. That is what it is. Now, when the protein is constructed, it can have one chain, or it can be multiple chain. Like we saw in carbohydrates, it may be one chain is there, many then branches are appearing. But in this case, we may not say branches are coming out of it, but many chains may be intertwined with each other, or one long chain might be folded within itself. All right. Now, where are these constructed in the cell? This polypeptide, this long chain, is manufactured by ribosomes in a process called as protein synthesis. I think we will have it next year in 12th. And these polypeptides that are there, they can be normal protein, they can form hormone, they can then form antibodies, or they can form enzymes. Now coming to the different structure. So these that are there, this is uh, like beads, all right, different amino acids joined together. So it's like if you have a pearl necklace and you can make it straight like that, so the pearls are attached, those pearls, each pearl represents an amino acid, so that is like a straight long chain. So that is a protein, that is one primary protein. Okay, so that is one protein. So it's like a straight thread is there, straight chain is there. Okay, that is your primary structure, that is called as primary, that is a straight chain, nothing much. So you take a simple thread in your hand, or like if you are having a long hair, so that is a long hair that you're taking in straight hair, that is a primary structure. Okay, that is what it is. It's like a simple straight chain, nothing much. So several hundred amino acids linked in a long chain, simple straight chain, your primary structure. Okay, now within this, the arrangement changes. Now you see that over here, cysteine and cysteine are held together by a bond or bonding called as disulfide bonding. Now, if I replace the cysteine with another amino acid, say for example, glycine or glutamine or aniline, something else than cysteine, then this bond will not take place. This bond will not take place. So automatically the structure changes. So the presence of the type of the amino acid complete, even a minute change, even a change in one amino acid alters the whole property of the protein. So you can see over here, giving an example, if I replace this, then this bond is not there. Automatically this will not be held this way. It will, yeah, this is like the sulfide is present, hence it is between them. Okay, so this statement that is there is very important to understand regarding protein. Okay, please make a note of this, either take a screenshot or write it down. Now, that is where, and that is the beauty of it. Okay, that is the beauty of it, that I can change the whole property of the enzyme and play with it by just changing a single amino acids and checking out what happens. The whole property changes. That is a beauty and a dangerous thing as well. If by chance a very good nice protein is there, for example hemoglobin, and I change one of the amino acid in that, the whole hemoglobin may just get destroyed and something else can happen. That is the case in a condition called as sickle cell anemia. Okay, I'm going ahead. Secondary structure. Now, in the case of secondary, what is there? In primary, what I said, there is a single thread, straight thread. Now you fold that thread, wrap that thread around it. So if you are making a spring out of it, okay, you have a straight chain and then you make a spring out of it or a coil, that is your secondary structure. All right. So if it is forming a coil-like structure, if you remember those old phone which had that cord attached to the base in a coiled form, like a spring, that is alpha helix. Okay, that is a helical structure. So that is secondary. So you take a straight thing, coil it up, that becomes a secondary. All right. They are forming due to the hydrogen bonding attached due to the closeness or proximity of the amino acids that are there among each other. Okay. So hydrogen bond between whom? Sorry. Hydrogen bond between whom? The carboxyl group and the amino group of the adjacent amino acids that are involved in the polypeptide. So alpha helix. Okay, another example is your Maggi noodle, the dry noodle that is there in the packet before putting it in the water and softening it. That is a helical pattern. So that is secondary structure. When you put it in the water and starts boiling, it loosens up, becomes straight. That is a primary structure. Yeah. Oh, there is no beta. Last point. You mean Niranjan? You mean this one or the primary structure? Oh, what is it saying is now this is this line. This line is primary. Now, to stabilize this, this may not be stable. It may break off. To stabilize it, it is forming a coil. All right. Now, this coiling is occurring. Why? Because of hydrogen bonding. Hydrogen bonding is between whom? This is an amino acid attached to another amino acid, attached to another amino acid. Okay. And then attached to another. Oh, yeah. Huh. What was I saying? Attached to another amino acids. Okay. So one, two, three, four. These two will form the hydrogen bond. Then another, another, another. Then like this. So what will happen after 4, 4, 4? Each fourth one will be involved in it, like fourth, 8, 12, 16, so on and so forth. Okay. Chalo. So this is a alpha helix. Then another pattern is the beta pleated sheet. Have you guys seen those asbestos thing? Okay. So basically, if you've seen certain rooftops where these are present, like tiles are rooftop having such kind of sheets, plastic sheets, asbestos sheet, yeah. Or on some nowadays on the terrace of the buildings, they've add those plastic sheets to prevent leakage and all, which has this folding pattern. That is a pleated. Pleats, okay, see, pleat means what? If you wear those formal pants or something, pleats are then folding, or girls might know about those sari folding thing. Pleats are made. So that is a pleated. Again, hydrogen bonding is occurring between the amino acids, but they are little loose compared to alpha helix. But then that is another pattern. Okay. Yeah. So this is there. Another thing about this is uh, these bonding that is there, they are kind of reversible. If I heat them or there is any change in the pH, will result in straightening out or loosening it. Okay. Classic example can be shown for curly hair. Okay, curly hair in the sense there are people who have like Maggie noodle like hair and all. They go for that heating hair iron or something that helps in keeping the head straight. You agree with me in that? Okay. Now it becomes straight due to heat. So now, as we saw, hair is made of protein. So straightening it, so it becomes straight. So that is where it is telling that bonds are temporarily sent away. Now, if that individual is washing the hair or something, later on that curliness comes back. So that is what's saying that it is, they can be broken with high temperature or pH change, but then pH change, for example, like chemical straightening, what people do with the hair. So it is permanently there as the chemicals are present, chemicals are being used, but the new hair that is coming in afterwards again turns curly. So anyway, so that is an example been shown about it. Just to make you, if you guys are able to hear me and understand it regarding the folding pattern. Okay. Then tertiary structure. Further folding. So what you do is you took a thread, you wrapped it. Okay. So now what is there? So this is a primary structure. When I fold this in one pattern, that is secondary structure. अच्छा मैं आता हूँ कुछ देर में. Okay, then when I further fold this among themselves in a particular way, then it is okay, how to explain this part? Okay, I have a very long hair, straight hair, so that is your primary. I make breads in that, that is secondary. I fold the breads and tie them up together, that is your tertiary. Does it make sense? Okay, yeah. Gift box ribbons. Yeah, gift. Yeah, Devangi, thank you for it. What I'm saying is that I have a very long hair. Have you guys seen the movie Rapunzel? Tangled. She has a very long hair, like 70 feet or something. Okay, that is not wrapped around. So that is primary. When she is folding it in one pattern, that is secondary. And she makes a fold over the fold, that is tertiary. Getting the idea of what is primary, secondary, tertiary structure? Okay. And Devangi, there's gift box ribbons. What you can say, the straight ribbon that is there is primary. When you like fold it or something in one pattern, that it can make curls if you hold it in a pattern. So that is secondary. And you take the curly ribbon together and wrap it around something, again fold it, then that is tertiary. Okay. I hope I got through to everyone. Then next is, now in this pattern, what is there? There are 4 types of bond involved in the making of tertiary group. Okay, for the tertiary, there is hydrogen bonding, there is disulfide bridges between cysteine, as we saw in the first slide. Okay, so disulfide occurs between the cysteine because it has sulfur in it. So when they are close enough, they will show a bonding. So it will lead to folding, more folding of the protein. Then ionic bond between the charges. Okay, then there is formation of the ionic bond. No. In primary, we don't observe any bond. You can say it's just a straight chain.